Okay, everybody, today's topic is protein synthesis. Remember, the main job of the cell is not to divide. The main job of the cell is to do whatever it's supposed to do. And the way it does those jobs is by making proteins. Now, kidney cells, brain cells, liver cells, skin cells are all going to make different proteins, right? But remember, every cell has a full copy of the DNA. So the process we're going to talk about is it's a two-step process, easy as 1, 2, transcription and translation. Okay, so just as a review, remember DNA codes for proteins. Those proteins control what happens inside a cell. Your DNA contains genes, which are the instructions for making those proteins. It's like a cookbook. Okay, so DNA controls which cell functions, right? It's kidney cells, liver cells, skin cells, brain cells. They all do different things by controlling which genes get turned on and turned off. Okay, two basic steps, like I said. First step, DNA is transcribed into mRNA. In step 2, mRNA is translated into a protein. Transcription, translation. Here's how I remember it. They both start with trans, but then C comes before L. So transcription comes before translation. Okay? This process is really important, right? DNA to mRNA to protein. This is like the central thing of genetics. Okay? So remember two processes. Transcription, DNA into mRNA. The little M stands for messenger, right? It's a little message that's going to be sent. And then translation, we take that messenger and we deliver the information and turn it into a protein. Okay, so some key players here. Sometimes students get a little confused. There are three kinds of RNA that we're going to talk about. Right? The first player is mRNA. The little M stands for messenger. That's going to actually carry the message from DNA to the ribosome. The ribosomes are actually made up of rRNA. The little R stands for ribosomal RNA. That's actually where the protein is going to be made. Remember, they're out in the cytoplasm and also in the rough ER. And finally, the last player, tRNA, the little t stands for transfer. That's going to carry the specific amino acids to the ribosome in order to put them together to make a protein. Because remember, proteins are made up of amino acids. Okay. Some differences between DNA and RNA. DNA, as we know, has a double helix structure. RNA is single-stranded. It's a lot smaller. And that's going to become important in just a minute. The sugar in DNA is deoxyribose. That's where the name comes from, DNA. The sugar in RNA is ribose. That's why it's RNA instead of DNA. And if you recall, in DNA, A pairs with T, C pairs with G. RNA is a little bit different. There are no T's in RNA. A pairs with U. So anytime you look at a strand and you see U's, you're looking at RNA. If you look at a strand and you see T's, you're looking at DNA. Okay, so let's take a look at transcription first. This is step 1 of the process, DNA into mRNA. Okay, now if you remember, DNA is in the nucleus. It's nice, it's protected, right? The ribosomes are out here. So the DNA can't leave the nucleus. It is too big, right? It's stuck in here. So what we need to do is make a little messenger for it to leave the nucleus, go out to the ribosome and deliver the message. Think of DNA being my desktop computer at home. I don't need every single file to come to school and teach a lecture. So what I do is I put it on my flash drive. My flash drive would be the mRNA. It can actually leave my house and go to school and then deliver the message. It's a temporary message. It's going to go away. So if I lose my flash drive, it's not too big of a deal because I still have my desktop computer at home, my DNA. Okay, so mRNA carries the info out of the nucleus to the ribosome, and we can actually deliver that message. Okay, so here's the steps. The DNA unwinds and separates, just like it would if it was going to replicate. Okay, so here the DNA, but not the whole thing, just the area that we care about, right? Just that particular gene sequence unwinds and unzips. Then the one strand, here's the strand of DNA, right? The coding strand, it turns into a template strand so we can start assembling our mRNA. And remember, there are no T's in RNA, so A will pair with U. A pairs with U, T pairs with A, G pairs with C, C pairs with G, and so on, right? Remember, mRNA is only single-stranded, so we're only going to have one strand. It's a lot smaller, so we can leave the nucleus. Okay, So we match up those nucleotides, we assemble that RNA. Once it's complete, the DNA will just zip back up and wind back up because we're done with it. And then we have our strand of mRNA. So here's our strand of mRNA. Now, something kind of weird happens. It's actually processed, right? There are some areas that we don't really know what happens or what it's used for, but they're removed. They're called introns. Those introns are removed, and then the exons, right? Exons are expressed. Those genes are expressed. So the introns are removed, and the exons are all pushed together and spliced up, right? It makes a very small, very nice, very complex piece of mRNA that can actually leave the nucleus. Okay? So introns get removed, the exons get spliced together, get put together, and they can actually leave the nucleus. Okay? And remember, there are no T's in RNA, so A will pair with U, right? So if this is your DNA transcript, this is your mRNA. Why don't you try it? So pause the video. Here's your coding strand of DNA. You make the mRNA strand. Remember, there shouldn't be any T's in your mRNA. So that's what you should have gotten. Okay? That's transcription. Now in the next video, we are going to look at translation. That's step 2 of the process of making proteins.